Our learning target for this lesson is I can describe how cellular respiration breaks bonds of food molecules and transfers the released energy to ATP. And your success criteria are that you can identify and describe the reactants and products involved in cellular respiration. We've already seen all of them. You'll see something really cool about that in a minute. Describe the structures of a mitochondria. More specifically, the three main parts we're going to focus on are the matrix and the inner and outer membranes. And then number 3, explain how a cell can manage to process energy with or without oxygen. So we're going to talk about 2 different types of plan Bs. Okay, so cellular respiration aerobic and fermentation anaerobic cellular respiration. Okay, so first, cellular respiration is the process that breaks down glucose, okay, and transfers that chemical energy into ATP. Who can tell me why does that cellular or chemical equation look familiar? Why does this right here look so familiar? Oops, if it's gonna let me write it out. Why does this look so familiar? Look at that compared to what we talked about on Tuesday. Why does that look okay? Because it reminds you of math. There's not a whole lot of math we have to do here. But this should look really familiar. Okay, water is familiar. Yep. But look at the equation as a whole. Yep, it has glucose. Compare this equation to the photosynthesis equation. Yes, Xavier, exactly. It is the exact opposite of the other equation. Okay? So photosynthesis was the opposite. We started with 6 CO2 and 6 water molecules and light. And we made glucose and oxygen. So we are literally just swapping them. Okay? So now we have glucose and oxygen that go into my equation. Those are my ingredients, those are my inputs. And then we're making carbon dioxide, water, and energy in the form of ATP. Okay? So I told you guys that this was going to be super important, this equation, and this is why. Because they're just inverses of each other. Now, super important to note that the main purpose of cellular respiration is to make ATP. Okay, so the main purpose of cellular respiration is to make ATP. What plants did in photosynthesis is they took carbon dioxide, water, and light energy, and they stored that light energy in the glucose molecules. Okay, they made their own paycheck. We don't have to make our own paychecks because we eat food. Okay, so we eat our energy paychecks. That's where our glucose comes from. Every single organism, even plants, go through cellular respiration. Okay? Remember back when we first started talking about glucose and biomolecules in Unit 2, and we said that glucose is your paycheck and you have to cash it at the bank to get ATP, and that's your your cash is you can do things with ATP, but you can't do things with glucose. Well, this is the process of taking it to the bank. So we're going to start with our glucose, we're going to break it down, and we're going to turn it into ATP, which we can use as energy. And our cells use that to carry out every other function that they have to do that requires energy. Okay, it can be used for things like active transport, it can be used for helping to make proteins, and so many other things. Okay, are you with me so far? We're pretty quiet in the chat today. Okay, good. Now, just like we said, photosynthesis takes place in the chloroplast, and we had to learn about a few different parts of the chloroplast. Cellular respiration happens in the mitochondria, and we have to know a few of the parts of the mitochondria. So let's look at that. So remember that the mitochondria, okay, is like the jelly-filled donut of the cell. So this whole structure here is your mitochondria, okay? Your mitochondria is free floating in your cell. And remember that the cells are full of the cytoplasm, okay? So the cytoplasm is outside of the mitochondria. The cytoplasm is the liquid or jelly-like substance in your cell that helps it keep its shape and kind of houses all of the organelles. So cytoplasm is outside of the mitochondria. And there's three main parts that we're going to talk about the mitochondria. There is the outer membrane, okay, which is this part around the edge. That's what keeps the mitochondria together. We have the inner membrane, which is this part here, okay? So it kind of separates the filling from the outside. And then the matrix is, I'll do this in a different color. The matrix is all of this space in here. Okay, so this is all the matrix. So if you look at this like a donut, the matrix is all of the jelly in the middle. Okay, that's your jelly-filled donut. Okay, so all you need to remember about this is there's an outer membrane that separates the cytoplasm from the rest of the mitochondria. There's the inner membrane, which houses the matrix. Okay. So there are three major processes in cellular respiration. Okay. The first one is glycolysis. And glycolysis happens in the cytoplasm of the cell. Okay? So glycolysis is the only step that does not happen in the mitochondria. So this happens in the cytoplasm of the cell, just outside of the mitochondria. And after glycolysis, okay, two different things could happen. Well, technically three different things, but we either have plan A, which is in a perfect world when you have lots of oxygen. So this happens when there's lots of oxygen to go around, okay? And when there's lots of oxygen, we're going to go through plan A. And plan A is going to take us through the Krebs cycle and the electron transport chain, or the ETC. Now, the Krebs cycle is not to be confused with the Kelvin cycle. Okay? The Kelvin cycle starts with a C, and that's photosynthesis. The Krebs cycle starts with a K and is in cellular respiration. So if we have lots of oxygen to go around, we are going through plan A. Okay? Yes, Krebs kind of sounds like crabs, like Mr. Krabs. That's a good way to remember it. Now, what do we do if we don't have a lot of oxygen available? So, if there isn't enough oxygen, okay, then we have to move to plan B. And plan B is going to be a type of fermentation, okay? And there's two types of fermentation that we're going to talk about, and that is lactic acid or fermentation. And the key thing to know about these is that both types of fermentation happen in the cytoplasm of the cell. So when we're on plan B and there's not a lot of oxygen available, we do not need the mitochondria. Now, different times that we would go through fermentation are like if you're working out. So if you are running or lifting a lot of things, or let's say you're helping somebody move and you're going up and down stairs and all around, like, you ever heard of feel the burn? Have you ever felt the burn? Either when you're working out or doing activities? Okay, so that burn that you're feeling is lactic acid. Okay? So lactic acid is what you are feeling when you're working out. It's a buildup of lactic acid. And that's because your body isn't getting enough oxygen to go through plan A. So it's going through plan B and it's producing lactic acid, which is the burn that you feel when you are working out. Um Now, the first thing sorry, I'm laughing at the chat. The first thing or the first step in both of these plans, plan A and B, the first thing to happen is glycolysis for both of them. So you have glycolysis, and then depending on how much oxygen is available, you're either going to plan A or you're going to plan B. Okay? And so as we go through each of these processes, we're going to learn the difference and why the Krebs cycle and electron transport chain are plan A and so much better than plan B. So, fermentation is a process that allows glycolysis to continue making ATP for the cell when there's little to no oxygen available. Okay? So, fermentation is really important because and we're going to go through the steps of glycolysis in a second, but when glycolysis happens, it produces NADH, okay, which is a lot like NADPH or NADPH. Um, it's just an electron carrier, okay? It's one of those carrier trains. And it also produces what we call pyruvate. And if there's a whole lot of pyruvate and NADH in your cell that's building up, your cell is going to recognize that and it's going to stop the glycolysis process. Okay, because you can't have that pyruvate isn't getting broken down anymore. And so if it stops the glycolysis process, then you're not making any ATP because glycolysis produces two ATPs. And so what fermentation does is it steps in and says, "Hey, let me take care of this pyruvate for you, and glycolysis is going to continue happening." And we're going to break it down step by step. So if that overview is a little bit confusing, it's okay. It won't be by the end of the lesson. So before we jump in and get started breaking these down, how are we feeling so far? Everybody with me? Any questions come up yet? Okay. So let's talk about step 1. Okay, step 1, this process is going to happen no matter what plan we're doing. Okay, so glycolysis is the first step. Now, remember when we were talking about glucose the first time, and we were talking about in unit 2, we broke down a lot of words, right? And said what they mean. Well, oops, lysis, okay, means to split. And this glyco is referring to the glucose, okay? So it's literally telling you in the name what the process of glycolysis does. And that is cutting the glucose in half. So we're going to go from a 6 carbon sugar to 2 3 carbon molecules. Okay? So we're taking this glucose that has 6 carbons and we're making 2 different, we call them pyruvates, okay? But it's just breaking this in half and then we have 2 different pyruvates. We get a net of 2 ATP and then we get 2 NADHs, okay? Now, again, your NADHs, we're going to be using, instead of NADPH, okay, we have NADH, which is your full train, and then we have NAD plus, which is your empty train. Okay, so these are just electron carriers. Now, what does it mean when I say net right here? What does this mean? And it's somewhere else on the slide. What does net mean? Good, overall. Okay? So, um technically the process of glycolysis produces 4 ATP molecules, but it takes 2 ATP molecules to cut this glucose in half. So if I'm using 2 ATP and making 4, that means that I have a net gain of 2 ATP. So that's why we just say that there's 2 ATP right here. So you need to know for this one that in glycolysis, we break down glucose and we get 2 pyruvates, 2 ATP, and 2 NADH. Now, ATP, just as a reminder, is our energy carrying molecule for the cell. Okay, that's our energy cache. And then NADH is just your electron carrier. Okay, that's our high energy electron carrier. That means that is your full train. Okay. And again, both cellular respiration and fermentation have glycolysis as the first step. So no matter what, glycolysis is step number 1. So once we go through glycolysis and we have those 2 pyruvates, what happens to them next? Because pyruvates are not a lot of ATP. Okay, we can't use those. Those are like a partial paycheck. So how do I get from pyruvate to a lot of ATP? Well, it depends. Is there oxygen or not? So, when we talk about breaking down words, we're going to break down a few more. Um, Aero, okay, this AERO means oxygen. Okay, so in aerobic, that means that oxygen is available. And so if oxygen is available, then we're going to go on with plan A. Okay, this is plan A. So aerobic oxygen. Aero is oxygen. Okay, that means we're going with plan A. This side over here is plan B. Okay, so let's break down this word. This AN means without. And again, this AERO, this arrow, means oxygen. So anaerobic means without oxygen. Okay? So that is no oxygen available or little to no oxygen available. Okay? And that is when cellular respiration stops and fermentation would start. Okay. And you're going to see CR a lot in this. So I just want to make a reminder to you that CR is cellular respiration. Okay. Questions so far? What questions can I answer? Okay. So I'm glad everyone is with me right now. So let's go to step 2. Okay, we are going to assume for the time being that we are going with plan A. Okay, so we're talking about cellular respiration. With cellular respiration, step 2 is the Krebs cycle. Okay, so remember that this is plan A. Okay, so we're moving on with the Krebs cycle. Now, we are only asking you to know what goes into the Krebs cycle and what comes out of the Krebs cycle. Okay. If you are curious as to the parts of the Krebs cycle, because it is kind of complex, there are a bunch of slides down at the bottom that says part 2 of the lesson. That's more advanced than we are going to go in this class. But if you're curious about it, you can find it down in the bottom few slides. Okay. So all you guys need to know for this is that the Krebs cycle, okay, breaks the bonds from the pyruvates to create carbon dioxide. And it occurs in the matrix of the mitochondria. Okay, so the matrix is your jelly-filled center of the mitochondria. Okay, so my ingredients for this, or my input, is 2 pyruvates. Okay, and they go in one at a time. So this cycle will happen twice. Okay, with my 2 pyruvates going through the Krebs cycle, I'm going to be getting 2 NADH. I'm going to be getting 2 FADH2s, which we'll talk about those in a second. I'm going to be getting 6 CO2s and 2 ATP. And anytime you see ATP, guys, I'm going to be outlining it in that like weird squiggle line, because if you've ever read like comic books or anything, that's like your pow, your bam, your wow, all of those things. Because ATP is your energy. So that's why I'm outlining them like I am. Now, Mrs. Stauffer, what the heck is a FADH2? I've never heard of that. That's okay. FADH2 is just like NADH, okay? They are high energy electron carriers. The main difference is that FADH2 is able to carry 2 hydrogens, okay? So this H2 means that this has 2 hydrogens. This H here means that that only has 1 hydrogen, okay? So it's just high energy electron carriers. This just means you have 2 different trains that are going back and forth and shuttling electrons and hydrogens, okay? So for this one, we have 2 pyruvates that come in, if I don't cross it out by accident. So we have 2 pyruvates that come in. We produce 6 carbon dioxides. We produce 2 ATPs. We produce NADH and FADH2. Now, we will ask you to keep track of the number of ATPs. You don't have to keep track of how many NADH and FADH2s we're using. Okay. Questions on the Krebs cycle. Okay. If you guys have questions, feel free to put them in the chat or the Q&A section, okay? If you have questions, it's important that we address them. So, assuming that we still have energy or still have oxygen, we have step 3. Okay, and step 3, again, this is still plan A. This is still plan A. Okay, step 3 is the ETC, or the electron transport chain. Okay, so this is where the magic happens. Okay, the electron transport chain produces a lot of ATP. So the energy, the electrons, okay, from NADH and FADH2 lose energy as they move down this electron transport chain. Okay, so FADH2 and NADH are dropping off their electrons and their hydrogens, and they're moving down this train. Okay, well, this is where oxygen comes into play and is super important. Okay, so oxygen is an input, it's an ingredient here. And this oxygen we get from breathing, okay, or plants and other organisms could have as a byproduct of something. Okay, so remember photosynthesis produces oxygen. So they have these oxygen and the electrons and the hydrogens that got dropped off because H pluses got dropped off too and moved down. They meet with the oxygen and they form water. Okay? And when they're building those bonds, the energy from those electrons making that bond produces 32 ATP. 32. So our first 2 steps, glycolysis and the Krebs cycle, each only produced 2 ATPs. The electron transport chain is like, sit back and relax. I got this. I'm the professional. And it gets out 32 ATPs. Okay? That's a lot of energy. What this is demonstrating over here to the side, yes, the electron transport chain is the overachiever. Okay, but we like the overachiever because that's what gives us the most energy. So looking over here, okay, these are my ingredients. My 6 oxygens or 6 O2s, NADH, and FADH2 are all coming into this equation, okay? And we're producing water and 32 ATP, okay? Really important. The oxygen captures the low energy electrons to form water. So that's what this is showing. So this down here is just showing you that the oxygen is combining with the hydrogen and the electrons, and it's producing water. Now, where does the electron transport chain take place? It does not take place in the matrix. This one occurs in the inner membrane of the mitochondria. Okay? So, where the Krebs cycle takes place in the jelly filled center, the electron transport chain takes place in the inner membrane. Okay? So, let's talk about aerobic, which means with oxygen, cellular respiration. With, I don't know why this says replace, with a lot of oxygen levels. Okay, so with high oxygen levels, I think this is supposed to be blank for us to write the replacement in. So with high oxygen levels, cellular respiration breaks down the pyruvates to create a total of 36 ATP. You need to know, hint, hint, wink, wink. Okay, hint, hint, wink, wink. You need to know 36 ATP, and you need to know how many ATP are produced at each stage. So, how many ATP do I get from glycolysis? How many ATP do I get from glycolysis? Good, 2. And even better, a net of 2. Yes. So I get 2 ATP from glycolysis. How many do I get from the Krebs cycle? How many ATP do I get from the Krebs cycle? Good, 2. Now, how many does my overachiever electron transport chain make? Good, 32. And 32 plus 2 plus 2 is a total of 36 ATP, which, if you couldn't tell, is super important. Okay. Gentle reminder for those of you just getting back. We are working on Wesley's question. You are not to put any answers in the chat whatsoever. Okay, so we're not giving any students any answers. I just wanted to give a little bit of a gentle reminder there.
It's like we're on the same wavelength. Get it? See what I did there? It's
Yep.
photosynthesis. Painful.
So, give me something in the chat to let me know you're back. Xavier, I'm glad you thought so.
I'd crack myself up if nobody else.
If you guys didn't know, we like to have fun. And we're often
Even at the expense of ourselves.
Yes. Okay, so this picture right here is such a good picture, okay? Which is why we're going to go over it for a second before we do, as a good recap, before we fill in a chart about cellular respiration, okay? So, first, I want to point out down here, we have NAD plus and we have FAD plus. So these are your empty trains. And then NADH and FADH2 are your full trains. Now, I had somebody ask in yesterday's class, I'm having trouble remembering what the empty train, is called. Like, how do you remember NAD plus? Well, if you know the full train of NADH, that train's job is to transfer hydrogens and electrons. So if I drop off the hydrogens and electrons and I'm left with an empty train, well, what is NADH minus the H from the hydrogen? It's NAD. And then how do I remember that it's positive? Well, electrons are negative. So if I take away a negative, I'm left with a positively charged train. So I have NAD plus. Same with FADH2. If I get rid of the hydrogens, I'm left with FAD. And if I'm getting rid of electrons, it's going to be positive. Okay? So I just wanted to make a note of this part down here for you. Okay? Now, we start with glycolysis. Okay? This is step number 1. Glucose comes in, and we get 2 ATP. We get NADH, which comes all the way down here to the electron transport chain. And then we get 2 pyruvates, which go right into the Krebs cycle. And reminder, glycolysis takes place in the cytoplasm. The Krebs cycle is in the matrix, so in the absolute center. The Krebs cycle takes in these 2 pyruvate. It's going to produce carbon dioxide and 2 ATP. Okay. Notice how we have this cycle in the center with our full trains going to the electron transport chain and then our empty trains going back into the Krebs cycle. Okay, that is just like in sorry guys, I lost my train of thought. No, you're good. So that's just like NADPH and ATP recycling in photosynthesis. Okay, so these guys have their own little cycle. They're just your shuttling trains. Okay. And so the NADH and the FADH2s from all of the processes end up here at the intermembrane or inner membrane to go through the electron transport chain. So all of the NADH and FADH2s drop off the electrons and my hydrogens. They go down the chain. They combine with oxygen. And this is where we get our water molecules and the big 32 ATP. Okay. And again, how many ATP do I get total from the cellular respiration? Good. 36. Okay. 36 ATP. Really important. Okay. So let's review this and make sure that we're all on the same page here. Where does glycolysis occur? Where does glycolysis take place? Good, in the cytoplasm. Oops. This here. Okay, in the cytoplasm of the cell. What goes into glycolysis? What's my one ingredient that I need for glycolysis to happen? Good, glucose. So glucose is my one reactant that I need. And what are my products? I have 3 different products that I'm getting from glycolysis. What are my 3 products that I'm getting from glycolysis? Okay. Yes, Serenity, but remember, I want the net total of ATP. So I only want to put down 2 ATP. That's okay. It just, if you remember, if you use 4 from that one, then your total number of ATP is going to be 38, and that's going to be incorrect. So we get 2 ATP, and then we get 2 pyruvate, and we get NADH. Okay. You're right. We do get 2 NADH. You don't have to keep track of the number of NADH and FADH2s. Okay. We're not asking that of you. So I'm going to get rid of that one just for now so it doesn't confuse everyone. Okay. So that is our glycolysis. Now let's talk about the Krebs cycle. Where does the Krebs cycle take place? Good, the matrix of the mitochondria. Oops, so let's do the matrix of the mitochondria. And I'm just going to write mitochondria over here so I don't have to do it twice. These are both in the mitochondria. So that takes place in the matrix. Okay. What is my one ingredient that I need for the Krebs cycle? Good. 2 pyruvates. Okay. And what are my 4 different products that I get from the Krebs cycle? What are my 4 different products that I get from the Krebs cycle? Everyone should have the notes in front of them, so feel free to scroll up and look at the photos. Look at the equations. Good. So the biggest thing is I get 2 ATP. I get 6 carbon dioxides, so I get 6 CO2. I get some NADH, and I get some FADH2s. Okay? Now, we're on to stage 3. So where does stage 3 take place? Where does the electron transport chain occur? Where does that happen? What part of the mitochondria? Good, the inner membrane. So the inner membrane. Good. Serenity, the mitochondria is not used in photosynthesis. Photosynthesis uses the chloroplast. Is that what you were saying, or did I misinterpret your message? That's okay. That's okay. It's good that we're bringing these up now so we can give reminders on them, because when you're learning both of them in back-to-back days, it can be a lot of information to take in. What are the 3 reactants or ingredients that I need for the electron transport chain? I need 3 things for the ETC to take place. What are those 3 things? Good. I need oxygen. And then I need my full trains. So I need my NADH and FADH2s. So I need oxygen and then my 2 full trains. Now, what 4 products do I get? Or what 4 outputs do I get from the electron transport chain? Good, Serenity. Yes, I get 32 ATP molecules, which is really, really important. What else do I get? Good, I get water. So I get 6 water molecules, so 6 H2O. And here's a hint. If I started with full trains, I'm going to end with empty trains, right? So what are my 2 empty trains? What are my 2 empty trains? Good. NAD plus and FAD plus. Good. So I'll give you guys a minute to screenshot this or make sure that you have this written down before we move on to talking about fermentation.
So we're looking at 25-ish minutes.
Okay, perfect.
I'd like to take a few minutes to address Wesley's question from the last session. I don't want to do it right now because we're right in the middle of cellular respiration, but
Yep.
Yeah, cool.
Perfect. I will move quick. So this is a nicer looking table than the one that I just made, but you guys do have this in your notes. And then let's talk about what happens when oxygen is not there. Okay. So cellular respiration stops, okay, when oxygen is not present. And the reason that is because if oxygen doesn't remove the electrons start to back up. Okay, so oxygen comes into play at the electron transport chain. So if oxygen isn't here, then these electrons basically get scared and they run right back into their trains. Okay? Because they can't be used. And so if they can't be used, they're going to go back to their trains because that's where they were last happy. Okay? They filled up the train, everyone was happy. So when they got dropped off, they needed to make another molecule. And if there's no oxygen, they can't make water molecules. So they go back into the trains. And that would mean that the electrons wouldn't be able to travel down to make any more ATP. And so because of that, we have plan B processes. Okay? So anaerobic respiration, remember this means without oxygen. So without oxygen, we get fermentation. So in little to no oxygen conditions, fermentation takes the pyruvates and turns them into something else without using or making any more ATP. So I want you guys to think back. If this happens after glycolysis, how many ATP total do I get from fermentation or from plan B? How many ATP am I getting from plan B if this takes place after glycolysis? So I'm including whatever I get from glycolysis. Good. I'm only getting 2 ATP. Do you see how this is not efficient at all? If with oxygen, I can make 36 ATP, and without oxygen, I can only make 2. Does everybody understand why cellular respiration is plan A? Why that's the ideal plan to go through? Okay, good. So, fermentation takes place in the cytoplasm of the cell. There is no mitochondria here. So the mitochondria only gets used if we have oxygen. Okay? And it's called anaerobic respiration because it does not require oxygen. What it does require is the electrons from the NADH that we already produced in glycolysis, and it's going to transform the pyruvates into fermentation products. Okay? Now NAD will be cycled between fermentation and glycolysis. So now NADH and NAD plus are in a constant cycle between fermentation and glycolysis until your body gets enough energy again and we go back to plan A. What questions can I answer right now on this? Right. I mean, if you die, you don't have oxygen. So it would take place until your body no longer has oxygen. Okay. So, why do we even go through fermentation in the first place if it's not making any more ATP? Well, it prevents NADH and pyruvates building up in the cytoplasm because, like we said at the beginning of the lesson, if NADH and pyruvate are just building up and waiting for your body to get more oxygen, they're taking up important space in your cells, and so your cells are going to stop glycolysis from happening, which means you're not getting that constant flow of +2 ATP, because even though it's not 36, it's still better than nothing. So fermentation uses up that NADH and it breaks down the pyruvates, which allows glycolysis to continue happening. So at least you're getting a couple of ATP. Okay. So there are a couple things that it breaks down in. Okay. So this is showing you an exercising muscle, which is going to have a lack of oxygen. And so you're either going to get lactic acid or you are going to get ethanol, which is um, alcohol. So we will talk about that in the next slide when we go through some of the important parts of each. Now, can fermentation occur indefinitely? No. Fermentation products like lactic acid and alcohol are actually toxic to your cells. So that's why you feel the burn. Okay. So it's fine for a little while. It's fine while you're working out or while you're, um, you know, having physical exertion, but it can't happen all the time. Okay. It doesn't work indefinitely. It will start to kill your cells. So let's talk about the difference between the two types of fermentation. Okay. Now, both of them start with, oops, let's see if it'll let me write. Both of them start with NADH and pyruvates. But lactic acid fermentation produces two things: lactic acid and NAD plus because we're using up that NADH. Alcoholic fermentation starts with NADH and pyruvates, and it produces, does anybody have a guess what alcoholic fermentation produces? Yep, alcohol. It also produces carbon dioxide and NAD plus. Now, lactic acid, we'll finish out lactic acid first, and then we'll come back to fermentation. So alcoholic fermentation. So lactic acid occurs in overworked muscles. And that's why you have the initial burn and soreness. And this also occurs in yogurt bacteria. This is what thickens the milk. So after this, who wants to go eat a big bowl of yogurt, huh? So, lactic acid fermentation is what thickens the milk in yogurt, um, and allows for yogurt to be made. Now, alcoholic fermentation, and then we're pretty much done after this. How many of you have ever baked bread or watched somebody bake bread from scratch? Okay, good. Well, alcoholic fermentation is what occurs in Single-celled yeast. Okay? Um, how many of you, when making bread, have seen when when it rises for the first time before you roll it in a lot of extra flour and let it sit on the counter to rise again, and you know how it kinda gets that smell and you see all the bubbles coming to the surface of your dough? How many of you know what I'm talking about? Those bubbles that you see in your dough when it's rising? Okay, those bubbles are actually carbon dioxide that are being released from the, um, chemical reaction that's taking place, okay? Or, yes, the spaces in your bread after it cooks, so once you've cooked your bread, um, it those spaces that you see or those, like, air bubbles, as we call them, or I call them often in bread, um, those are actually where carbon dioxide were. So that's carbon dioxide being released from the yes, they are, bacteria farts, basically, or yeast farts. So, this also happens with brewing yeast, which gives us alcoholic beverages, and baking yeast, which we use for rising breads. So when you've baked or made bread with yeast, you know how you often have to activate your yeast first, where you mix it with some warm water and some sugar sometimes? That's because yeast are organisms, and you are kick-starting the alcoholic fermentation process before you mix that in with the rest of your ingredients to make your bread. Okay. Questions on fermentation? Okay. Here's a nicer looking chart than what I wrote, but again, that's in your notes. And so I want to see where you guys are at with this lesson.